Good morning, everybody, and welcome to the fifth webinar of the six-part webinar series Towards a Low Emissions Future, which is being run by the New Zealand Agricultural Greenhouse Gas Research Centre in partnership with the Ministry of Primary Industries. My name is Sinead Leahy. I am from the centre, and I'll be helping to facilitate the session this morning. And it's a great session this morning. It's all about the science, and we've got three great presenters coming your way. For those of you who haven't been at one of our webinar series this year, how it all works is each of our presenters have pre-recorded a presentation for us. So we'll hear from each of our speakers. After each presentation, we'll take 1 to 2 questions. If you want to add a question, if you just look to the bottom of your screen, you should see a Q&A panel. If you click on that, you should be able to write some questions. If there's a particular question that you like, you can hit the upvote button, and that lets me know that it's a question that you'd like to have answered online. All of our presentations this morning will be made available on the NZAGRC website. Also, I apologise in advance, we may not be able to get to all of our questions, but the NZAGRC will be following up with all of our presenters to get answers to any of the questions that are asked this morning, and we'll be putting them on the website in due course. Now, I realise time is very precious, so I'm now going to introduce the very first speaker, Dr. Nicola Walker. Nicola is a principal investigator from the international company DSM Nutritional Products. She's no stranger to New Zealand, having spent some of her research career here in New Zealand at Ag Research in Palmerston North, where the centre is actually based. DSM Nutritional Products have been doing some very impressive work in the methane inhibitor space, and we're delighted that they've been able to contribute to our webinar this morning. So let's hear from Nicola Walker on their methane inhibitor, Bovar R3NOP, and let's get the latest updates. Let's roll it there, Kate. Thank you.
Hello everybody, I'm Nicola Walker. I'm the principal scientist on the project Clean Cow working for DSM, where our goal is to reduce enteric methane 1 burp at a time. And this is like one of the coolest projects ever because I get to talk about cows, which I love massively. And I also get to talk about burps. It's awesome. So the talk today is going to be on methane inhibitor strategies for pasture-based farming systems. So if we actually look, there's a lot of work is being carried out globally to try to reduce enteric methane from ruminants. Now, there are 4 different options to do this. There's been a lot of work and research in the development of a methane vaccine program. There is also the possibility to select for low methane emitters through animal genetic selection. There's a range of different nutritional and dietary strategies that can be used. These can include fats, lipids, or increasing the amount of concentrate in the feed. And then the topic that I'm going to talk about today and really focus on is the use of methane inhibitors. Now, these are particularly important, perhaps, because they have the greatest potential to have a positive impact on methane emissions. So the inhibitor I'm going to focus on today is a product called Bovar. The name for this is coming from a combination of bovine and air to get Bovar. Although most people in the scientific community would know this as 3-NOP or 3-nitroxypropanol. Now the history of the development of this inhibitor has taken since 2008. Now, trying to identify inhibitors for methane is not a new thing. This has been a topic of research since for the last probably 50 years. And people have looked at the different dietary strategies, the different additives that could be used, breeding or genetic selection, and of course the vaccine program. In 2008, however, the decision from DSM was to start a whole new project called Clean Cow. And this was driven primarily from the drive to reduce methane, which is an important greenhouse gas. Now the story continues, it started 2008. I think DSM probably took a completely different approach to some other people. They used in silico modeling, which was still quite new at that time, and was being used by the pharma companies to develop new drugs. Through this, a variety of different molecules were identified, and in April 2010, the first grams of Bovaer were produced for the first time. Since then, there have been a large number of different studies that have been conducted in a whole range of different ruminants, and ranging from sheep, goats, dairy cows, beef animals, all the way through in a range of different diets. To the point where we're at now in 2019, we just recently filed for European registration. Now, this journey has been a long process. It's been a very interesting process. I only joined the team 2 years ago. I was very excited to join the team. And it's involved a huge amount of collaboration between a number of different research groups throughout the world. And we've actually produced in that time over 30 peer-reviewed publications, which have shown that the Bovaer consistently reduces enteric methane production for dairy animals, beef animals, goats and sheep. If we actually look, cows make methane. They are not actually responsible for making the methane. It's a process from the microbes that live within the rumen. This is an end product of fermentation. This particular group of microorganisms, called the archaea or methanogens, take CO2 and hydrogen and convert this into methane. And this is burped out by the cow. Why the interest in methane? It has strong global warming potential, 28 times that of carbon dioxide, but it has quite a short half-life. So anything that you can do to reduce methane can start to pay off pretty much right away. The amount of Bovaer that needs to be fed to the cow daily is a very small amount. It's a quarter of a teaspoon. This amounts to about 1.6 grams of the Bovaer per day. And the result is generally we find on average a 30% reduction in methane. Starts to work immediately. And all the research that we have done has shown that it's safe for the consumer, it's safe for the animal, and it's good for the planet. So very much the way it works is that the Bovaer targets one specific enzyme of the archaea, the methanogens, and inhibits it. So less methane is generated. So here we have a really kind of like awesome picture that just shows the target point. So the target is methyl coenzyme M reductase. This is the last step in the methanogenic pathway. You only find this enzyme in methanogens or in the archaea. And the work that was done using in silico modeling was able to identify the binding site for the substrate for methyl coenzyme M and block it. So as a result, we inhibit the formation of methane or slow down the formation of methane. The mode of action is well understood and documented. As I said, highly specific. It works by blocking the methyl coenzyme reductase and it inhibits methane formation. When it acts, it's actually broken down. The Bovaer is broken down to its two constituent compounds in the rumen, which is 1,3-propanediol and nitrite. This involves a catalytic reaction. The nitrite is then used again in a further reduction process in the formation of nitric oxide. And basically, the Bovaer gets broken down through its own mode of action and is metabolized in the rumen. The molecular details of the mode of action, this also, because it's actually broken down and metabolized, it doesn't kill the archaea, it just kind of slows down their activity, inhibits the formation of methane, and as a result, we think that adaptation is unlikely to take place. And we've run a study now running for 1.5 years on a dairy farm. We have seen no adaptation occurs. So, because we're not putting selective pressure on the microbes by killing them, they don't try to find a way around it. So as a result, we've not seen any adaptation occur. It's fully reversible. As soon as you stop feeding the Bovaer, methane production just goes back to where it was before. So it doesn't kill the microorganisms, it just slows down their activity. To date, we've done 12 dairy studies that have been published that show this consistent effect of Bovaer in dairy cows. This has mainly been done in PMR or TMR fed animals. This is more like a continuous feeding effect. So by having the Bovaer in the feed, we are matching the rate of fermentation, the formation of the methane, and then we inhibit it. The mean value of methane reduction across these different studies is around about 30%. And this value actually ties in well with an independent meta-analysis that was carried out by Jan Dijkstra's group at Wageningen, where they estimated around about 39%. It works in all stages of lactation. We find that the dose response is linear. So the higher the dose, the greater the reduction. And for us, at least in Europe, we're looking at an inclusion rate of between 60 and 100 ppm or milligrams per kilogram dry matter. And in fact, out of the studies shown there on the left, the last 10 most recent trials have been run at the lowest dose. And we're now currently extending that in new studies to investigate the range of different doses and to understand dietary interactions. So overall, we've done 39 different trials that have been conducted across the world, ranging from North America, Latin America, Europe, and even in Oceania. Now, for the studies in Oceania, it's of course a very special system. It's a pastoral-based system. So, we've had to actually do quite a bit of work to understand what impact feeding Bovaer in a pastoral-based system will have. So if we actually look on the left-hand side, we have a graph showing the methane production in animals fed a TMR-based system. On the right-hand side, we've got a graph showing the effect on pastoral-based systems, so in cut and carry grass. On the left-hand side, in the TMR-fed cows, we have the 3-NOP in red versus control cows in a bluey-green color. We have animals were fed once a day. The Bovaer was included in the TMR. And we can see that there's some gaps there which coincide with milking when animals were removed. Methane was measured using green feed systems, and in this case, the methane yield, or grams per kilogram dry matter, inclusion of Bovaer led to a reduction of 29%. On the right-hand graph, this was done at Ag Research in New Zealand. Here we're looking at methane production in a 24-hour period. Cows were fed cut and carry grass. And the Bovaer was either mixed with the pasture and fed continuously, similar to what we would have in a TMR-based system, or it was mixed in a supplement which was fed twice a day. And that is in the green line. So control, blue line, green line, twice a day feeding, red line, continuous feeding in the grass itself. With the pulse dose, or twice a day feeding, we see a 48% reduction in methane yield. With the continuous feeding, we got a 61%. Again, this was measured in respiration chambers. Carrying on from that study, we also did some further work, again with Ag Research, looking to see what the impact was in animals fed the Bovaer in different systems or different trial setups. In the first study, that's the one that is just presented earlier, just so that you can get familiar with how we present the data. We have the methane profile in the blue line. The red line coincides with hydrogen emissions. The green triangles are when the Bovaer was fed or supplemented in the twice a day feeding. And you can see at each point, Bovaer enters the rumen, we inhibit methane, we see a spike in hydrogen. So as I said, methane reduction was reduced, the methane reduction was by 48%. Figure 2 below, in this case, cows were put in the chambers. They were fed cut and carry grass at 8 a.m. in the morning and 4 p.m. in the afternoon. The cows were fed Bovaer at milking. and they had no access to feed for 1 hour. And the reason this was done to mimic the time taken for animals to walk back to pasture. So basically the rumen was on a lower fill, if that makes sense. In this case, we find that methane emissions were only reduced by just over 5%. And this shows that really that we need to start thinking about developing a slow release form and testing it. This led to 2 other studies. In phase 1, we looked at a range of different slow release prototypes. There were 12 different prototypes tested. This was done in 16 cows in a Latin square arrangement. Unfortunately, due to seasonality, this was done, animals were fed hay and concentrate rather than cut and carry grass. It was winter time, so we had difficulty supplying the grass. But in this setup in figure 1, the standard form of the Bovaer 3NOP was fed at 8 a.m. when animals were first given feed. In the afternoon at 4 p.m., we have one of the test formulations. Then the second day at 8 a.m. they were fed a placebo, and then again 4 p.m. they were fed the test formulation. On the first day, the test formulation was a half dose. On the second day, the test formulation was a full dose. And you can see the reduction in methane of the blue line with the spikes and peaks of hydrogen that occur at the same time. So 3 prototypes gave promising results. One formulation that's shown in the figure gave a slow release or persistence effect, which lasted for 10 hours, resulting in a methane reduction of 26% compared to 10% when it was fed in the normal form. This was then, we were then repeated the experiment in phase 2, except this time we used cut and carry grass. We did a little bit more tweaking on the different forms that were being tested. We had 9 forms that were tested in 12 animals that were fed cut and carry grass. Out of this, we also used a much lower dose, only 1.1 gram per day of the Bovaer, and we saw persistency of up to 7 hours, which gave methane yield reduction of up to 26%. So the process we're in the moment, we have some longer duration studies that will be performed with the best 2 candidates. So just to conclude, there are several different methane mitigation strategies that are being investigated by research groups across the world. Bovaer, or for the scientific community known as 3NOP, is a methane inhibitor. It's been proven to be highly specific in targeting the methanogenic archaea and efficacious in reducing methane emissions across different ruminant species fed different diets. Currently, the status we're at for developing forms for pastoral-based systems that are found in either New Zealand or Australia or even parts of Europe, we are developing these new forms and testing them so they can be used in these more extensive-based systems. Further research is going on to the use of Bovaer is currently ongoing to maximize its use in pastoral-based systems, and so that we can actually understand a little bit more the dietary interactions, dose rate, and the impact this can have on methane reduction. And just finally, I'd like to thank everybody for paying attention to this talk, and most importantly, I'd like to thank all our research partners. We've got many of them, for all the work that has gone to contributing to this great project. Thank you.
Thanks, Nicola. So lots of questions coming in, but maybe the first one that we might take is around cost. You know, do you have any comments on the cost? Someone has asked the question: has there been any analysis to look at the cost effectiveness of supplementing 3-NOP in a dairy system? And there's also been a number of questions as well as around the availability. You talked about it being registered for Europe. How long do you think it'll take to get registered there? But equally, what about New Zealand? Some questions around the availability in New Zealand. So, Nicola.
Okay, so if I start off, never ever actually ask a scientist how much anything costs. But what I can say is it is cost effective, and we're probably looking at return in terms of return on investment of at least 1-to-1, and that's just due to some of the differences we see in milk composition. Some of the we see some small reductions in dry matter intake on certain diets, etc., which more than adequately pay for the product without even taking into account carbon credits. So you know, it's one of those things where it's not going to be astronomically expensive. In terms of registration, we filed the dossier in the EU. For those of you not familiar with that process, that can be anything from 18 months to 2 years to get approval. Where we are currently at the moment, we're just waiting for some questions and in the process. And so we're estimating that it will probably be 2021 before it will be on the market in Europe. For New Zealand, it's kind of a strange situation. There isn't actually a category at the moment for methane inhibitors. So the current ruling we have is we could sell it today, but you can't make any claim on the methane. So that makes it a little bit tricky. So until there's new legislation brought into place, whereby there will be a category for feed additives that have a positive effect on the environment, we can't actually start to make any methane claims.
Okay, thanks, Nicola. I'll try and sum up a couple of the questions. Again, there's a couple of questions coming in around, you know, does it affect productivity? What about dry matter intake? And then also, you know, some questions coming in around, you know, how much work has been done around the market acceptability of, you know, 3-NOP, you know, methane inhibitors, you know, they could get a bad vibe. Have DSM looked at the market acceptability of their product?
Yeah, so that's one of the key things that we've done, is trying to understand, you know, how this will be more or less paid for. Indications are that on consumer surveys, that people are very interested to have green label type products, and they're willing to pay a little bit extra. Talking with some of the dairy processors and people throughout the value chain, they're interested to have green label products, which they feel will help. They also, with some of the large processors, they have the capability, of course, to incentivize their farm suppliers, because they're going to be able, they're large enough to actually make claims for the carbon credits and do carbon credit accounting. If we look as well in terms of, at least within certain parts of the EU, there will be CAP or sort of agricultural policy payments, which will also incentivize farmers to reduce their carbon footprint or hoofprint. So there is going to be incentivizations there. In terms of performance, we're currently, what we can see from the data that we have in terms of productivity, we can see a 5% improvement on feed efficiency. This can either be driven by an increase in energy-corrected milk or driven by a decrease in dry matter intake. The way the product works is we tend to channel some of the hydrogen, which would otherwise have gone into methane, into propionate. The problem when you start to accumulate propionate in the rumen is that it signals satiety to the cow, and then you have a slight decrease in dry matter intake. Other things we've seen in terms of performance is an increase in fat and protein. We've looked at processability, so you know, is there any impact on the quality of the milk? We've looked at taste panels with yogurt, cheese, and other dairy products, and found, in actual fact, for at least the yogurt, we find that the product tastes more creamy. This is because we tend to have a slight change in the fat globule size. We find a slightly higher fat and protein content.
Brilliant. Thanks, Nicola. I'll just do one more question because it's the top one. It's just basically, is it detectable in milk? And a few questions around residues. So I don't know if you can quickly answer that.
Quickly, I can tell you that 3-NOP, we don't find any residues of the 3-NOP. It's completely metabolized in the rumen. and we've not found anything in the dose rates that we're using. We've not found anything in the milk either. So everything is below the limit of quantification. You can always run the risk when you super increase sensitivity on testing, etc., that you might find something, but we're already down at the parts per billion range. So really, you know, yeah, we haven't found anything there.
Brilliant. Nicola, thank you very much for your contribution this morning. Due to time, everyone will move on, but as we said, we will work with Nicola to answer all of the questions that you've asked us this morning. Next up, I'd like to welcome Dr. Peter Janssen. He's a principal scientist at Ag Research. I've known Peter for many years and worked with him, and it's fantastic that we have him on the webinar series this morning. Peter works across many of the ruminant methane mitigation research programs in New Zealand, and this morning he's going to be talking about the methane vaccine. Now, the methane vaccine is one of those technologies that we'd all really like to have in our toolbox. So let's hear from Peter around the latest updates of where the science is at when it comes to a methane vaccine. Roll it there, Kate. Thank you.
Hello, my name is Peter Janssen. I'm a scientist at Ag Research, and today I'm going to tell you about our program to develop a vaccine to try and reduce methane emissions from ruminant animals in farm systems. So everyone's aware of vaccines at the moment, of course, because of SARS-CoV-2, the virus that causes COVID-19. I'm going to tell you about our work to develop a vaccine against methanogens, why we're doing it, and where we're going. So at the end of the talk, really, I want you to understand why we want a methane vaccine. I'm trying to tell you how it works, and I'm going to give you a bit of an update on how we're getting there. So why do we want a vaccine? So obviously we want to reduce the amount of methane that's being formed in farming systems from farmed ruminants. The attractiveness of a vaccine is that it's a low frequency intervention. It doesn't require change by the farmer in their farm system. Vaccination's already an accepted farm practice, and use of a vaccine doesn't result in chemical residues in the products that are being sold on our local and international markets. Vaccines are also applicable to all farm ruminants, which again adds to their attractiveness. Now, of course, it's important to think about the feasibility of it as well as the attractiveness of it. There were attempts made by CSIRO to develop a vaccine against rumen methanogens, and in parallel also against protozoa in the rumen, and these did not work. However, there have been successful vaccines produced that do target processes that occur in the rumen, which is where the methane is formed. One of these is a vaccine that targets microbes that are responsible for acidosis in grain-fed animals. And also there are reports of a vaccine that successfully target the urease enzyme, again, a process in the rumen. So what are these methanogens, these microbes that are forming the methane? These are the ones we're trying to target. So the methanogens are a group of microbes that belong to the archaea, which are distinct from bacteria and also distinct from eukarya or eukaryotes. Eukaryotes are where we belong, humans, also animals and plants belong there as well. So the archaea are evolutionarily distinct from the rest of the rumen system, that is, the bacteria that are degrading the feed, which I'll tell you about in a minute, and also the feed itself, the grasses or plant material, and the animal, and also from the human consumer. So this panel here shows some methanogens. These are these blue dots or slightly oval looking organisms. Each one of these is a single organism of a methanogen, If you imagine a blue jelly bean in your hand, that would be similar to a methanogen cell, but about 10,000 times enlarged. So the blue color actually comes from part of that evolutionary distinctiveness. And that evolutionary distinctiveness also creates targets that we can use to try and reduce their activity in the rumen without upsetting the whole function of the rumen. There's more than one species. There are multiple species of methanogens in the rumen. So a vaccine has to try and target a significant part of this methanogen community rather than, say, like a COVID-19 vaccine, which is really targeting one strain in one virus species. Anti-methanogen vaccine needs to target multiple strains in multiple species. While there are multiple methanogen species in ruminants, the good thing is that the same species occur all around the world, regardless of whether it's in cattle or sheep or whether the animals are being fed grains or grasses. So that means that a methanogen vaccine should be widely applicable globally to reduce methane emissions. These methanogens dwell in the rumen, which is our colloquial term for the modified forestomach of ruminant animals, which consists of actually the reticulum and the rumen and the omasum. We just use the word rumen for simplicity. And this is where the methane is formed. These animals, these ruminant animals, they can feed on plant material that humans cannot use. And with the aid of this complex microbial community that lives in the rumen, these feeds, these grasses in particular, are fermented to products that are useful to the animal. So there's this complex community of bacteria and protozoa and fungi in the rumen. They ferment the ingested feed and they produce volatile fatty acids, which is the major component of the animal's nutrition. And also, in addition to that, hydrogen gas, H2, is formed as a byproduct. And the methanogens grab onto this opportunity to use this waste product because they can grow using this and convert it to methane. And this methane is eructated out, belched out by the animal into the atmosphere. So how are we going to stop this with a vaccination? Well, vaccination consists of antigens, which are components of the organism that you want to remove out of the system. And these antigens, when are vaccinated into the animal, stimulate the animal's immune system, which results in the formation of antibodies. Now, these antibodies that we're trying to produce are secreted into the saliva of the animal, and this saliva then enters the rumen as the animal swallows. So a cattle animal produces over 100 litres of saliva a day. So there's a plentiful supply of saliva carrying antibodies going into the rumen. And if we have developed the right antigens that induce the right antibodies, these antibodies will bind onto these methanogens and inhibit their function and so reduce the amount of methane that they're forming. So antigens are parts of unwanted organisms, or the organisms that we want to eliminate from our system, and they can be dead or weakened whole cells, or viruses for that matter, if you're trying to target a virus. And this was the approach that was tried by CSIRO, and we've also tried this at AgResearch. So you can get antibodies if you vaccinate with dead cells, and those antibodies can have an effect against methanogens in the test tube. But we haven't been able to get methane reduction using this approach. But there are other ways of developing or selecting antigens for a vaccine. They can also be a more defined part of a cell or a virus for that matter. So everyone will have heard about the so-called famous spike protein of the COVID-19 causing virus. This, of course, is the hot antigen target for anti-COVID-19 vaccine. So methanogens also have surface antigens that could be used in a vaccine. Here's a methanogen cell just showing it in relationship to COVID-19 causing virus particles so that you can get an idea of the size. And methanogens have hundreds of different potential antigens on their surface. So there's certainly no shortage of potential targets for developing a vaccine against methanogens. So why do we think that a vaccine targeting methanogens might work? Are there sufficient antibodies produced in the saliva? We've made studies to investigate this, and we know that there are sufficient antibodies produced if you vaccinate to target every single methanogen cell in the rumen. So there are sufficient antibodies. If we produce antigens that are only a component of a cell, do the antibodies that are produced bind onto the very specific target that we're aiming for? And we've shown that that is the case. If we take a part of a methanogen cell and we vaccinate with that, we can show that the antibodies produced target that specific component on the methanogen cell. And they do that on the whole cell. So it doesn't have to be a busted up methanogen cell. It can actually find that on the surface of the methanogen cell. So there's nothing obscuring the antibodies from accessing those targets on the methanogen cell. And importantly, this happens in rumen fluid, because there might have been something about the contents in the rumen that stopped antibodies binding, but that's not the case. And we also know that the antibodies that are produced can have effects on methanogen cells. So it means that when antibodies bind, they do something to the methanogens. And we have some experimental evidence of that. So antigens can elicit antibodies that have impacts on methanogen cells. It's shown in this example here. The top panel shows cells that have been treated with antibodies raised against an ineffective antigen, and the bottom panel shows how an effective antigen can cause cross-linking of the cells, which we call agglutination. So it's having an impact. In this middle panel, again, an ineffective antigen, the antibodies result in cells that have normal shape and size, but below it you can see cells that have been treated with effective antibodies from an effective antigen, and they've changed the size of those methanogen cells. So they're having some impact on the biology of the cells that's causing them to change size. And also, sometimes we see that antigens can actually result in a reduction of methane formation in a test tube. So while it's only a test tube, these results all show that you can get an antibody impact against a methanogen cell, which gives us the promise that it could reduce methane formation in the rumen. So the other part of this are the universal antigens. We've said that we need to target methanogens from multiple different species. Well, right in the very early days of this program, the PGGRC initiated the funding to allow us to sequence genomes of methanogens, and that's enabled us to look across the genomes of multiple species and identify that there are many components shared across the different species of methanogens. We also have experimental evidence that there are universal antigens. We've vaccinated sheep with just a single strain of methanogen and then shown that those antibodies bind right across the full range of the target species. So they're finding their antigens on different species. And also importantly, this happens in rumen fluid. And it's repeatable with different strains of methanogens, so there's a lot of cross-reactivity, which is very important if we want to try and develop a vaccine to target multiple species. So we do know that there are universal antigens. So at the moment, we're in the process of selecting antigens for a successful vaccine. We've identified nearly 400 potential methanogen antigens that could be used. We need to find those amongst that lot that actually inhibit the growth and the function of methanogens in the rumen. And that's what we're doing at the moment. We're screening to identify the ones that could be a part of an effective vaccine. So this is where we're at the moment. I think this is the really exciting part because we're shown that it can work. Now we just got to make it work. So this work has been carried out by a team of scientists at Ag Research led by Dr. Neil Wedlock from 3 of the research teams at Ag Research. The research was supported from the very beginning by the Pastoral Greenhouse Gas Research Consortium, which is supported by New Zealand's pastoral farmers and their organisations, and by MB, and also supported now by the New Zealand Agricultural Greenhouse Gas Research Centre, which is funded by MPI and MB. And we've also received support from the New Zealand government as part of its support for the Global Research Alliance. And we don't just do this all in a vacuum. We have a panel of advisors, which we call the Methane Vaccine Think Tank, which gives us feedback on our ideas and also ideas on new steps and new processes that we can take to expedite the development of the vaccine. Thank you very much.
Thanks, Peter. That's awesome. Some questions that come in, and it's that the first one really is, when did you first start work on the vaccine on methane production? And I guess that leads into, the age-old question, how long is it going to take you to get a vaccine, to market? It's obviously a technology that's most wanted, but obviously you've highlighted to us the scientific difficulties when it comes to a technology like this. Would you like to address that?
Yeah, so actually I haven't been at Ag Research, I've been at Ag Research for 13 years now, and the research started a little bit before I arrived. The initial attempts were to repeat the work that had been done in Australia, and that was unsuccessful. And then we really probably at a decision point: do you dive deep into the science or do you give something a go? And there was a bit of a double-pronged approach at that point. There was a lot of work done trying to isolate the methanogens, because at that time there was very little known about them. In fact, there were major groups of methanogens we didn't even know what they did in the rumen. They weren't even sure that they were methanogens at that stage. So there was a whole lot of work done on genome sequencing and trying to understand the methanogens themselves. And then in parallel, there were attempts made using some sort of obvious things, which obviously in the end didn't work, otherwise we would be a lot further on. So around about 2015, we decided to sort of science it a lot more, and we've gone into a much more, I suppose, more fundamental approach, took a step back and really tried to understand what wasn't working. And was there a reason why it might never work? And I think we've shown now that all of those reasons that we thought that might be barriers don't exist. They look like there's no theoretical reason why it wouldn't work. So we're now moved on using all of that information that we've gathered over that time from those genomes and all those organisms that we've isolated and studied, now to develop the vaccine by finding the right antigen or antigens to make a vaccine that will work. Once we've done that, and really that's the key, you've got to find that antigen. We've been told that it's something like 5 to 7 years after that to get your vaccine out there. But the unknown is that, how long is the piece of string, is how long will it take you to get that right antigen in the vaccine, to put into the vaccine.
Thanks, Peter. Some questions there about how you anticipate that it might work. Will it be lifelong? What level of methane reduction would you expect from something like a vaccine? What happens if you take out a particular subset of microbes that are crucial to the rumen? Any thoughts on that, Peter?
Yeah, so these are things that we obviously think about a lot. We suspect, until you have a vaccine, you can't answer a lot of these questions. So, you have to bear with me a little bit of speculation and the transfer of knowledge from other systems. We would expect that it might have to be an annual booster. We also think that, we know that the group that we're targeting, or the methanogens we're targeting, the ones that cross-react to these universal antigens, make up about 70% of the methanogens and are probably responsible actually for about 70% of the methane, more or less. It's about how effective those antibodies are. Do they slow them down and reduce their activity by half? So we might get 35%, or do they take them out completely? The question about will other ones pop up, that's why we're going for these, looking really for universal antigens. And as we've shown, we know they're there. Just got to identify which ones they are, because obviously you are concerned if you just take out one species, the others will just grow up and take over. But there's two major groups of methanogens in the rumen, broadly speaking, and they cannot proliferate into each other's niche space, as it were. You know, to take over the other one's roles. So we're not worried about that.
And just one more quick one, Peter, it'll be around, you know, cost. Obviously, you know, what's the cost of going to be for someone out on a farm who would want to use this type of technology? Do you have any indication on that?
No, I don't have any indication of that, except we don't expect a methane vaccine to be different in price to other vaccines. I think what's going to be very important, and the question was asked before about how much methane knockdown there is, that's that trade-off. Obviously, it depends on things like the carbon price. If it's too small an effect, you won't be able to get a vaccine that's economic. So that's about all I can say at this stage.
Brilliant. Thank you very much, Peter, for your contribution this morning. So our final speaker now, I'd like to introduce Dr. Suzanne Rowe. She's a senior scientist at Ag Research, and if you really want to talk to someone about animal breeding and low emissions research, then Suzanne is the lady to speak with. Animal breeding, you know, it's obviously a technology we're very familiar with. It's a practice we're used to. So let's hear from Suzanne around the latest updates around how animal breeding is going in terms for a low emissions phenotype. Over to you, Suzanne.
Hello, my name is Suzanne Rowe, and I'm going to talk to you today about the rollout in a breeding scheme for changes in enteric methane emissions in the New Zealand national sheep flock, and a few learnings and how we might take that forward, not only on a national scale in sheep, but also in other species such as cattle. So the challenge we face as livestock breeders is that 80% of the methane that's produced in New Zealand comes from enteric fermentation, and that's where feedstuff in the ruminant gut is broken down by microbes into an energy source for the animal. And during that fermentation, during that breakdown for energy, hydrogen and carbon dioxide are produced, and they're combined with bacteria into methane or CH4, and that's emitted or eructed from the gut as a waste product. So that the challenge that we face is that the animal is efficient and can still break down that feed, still create those energy sources, but that we actually reduce the waste product or the methane that's being produced. So this is a decade on. And I'm going to talk a little bit about the science project first that underpinned this. And basically what happened was that national flocks across New Zealand, so representative of the entire national flock, 1,000 animals were taken and they were measured through respiration chambers over a series of weeks, months and years. And the top and the low emitters from these 1,000 animals were grouped. So we have two methane selection lines. So that's 100 sheep that were the highest methane emitters and 100 sheep with the lowest methane emitters. And these were kept. They were kept in exactly the same conditions, but each year they were bred. The lowest were bred to the lowest and the highest to the highest. And the idea was to see whether actually they retained that methane status, whether the high stayed high and passed it on to the next generation, and the low stayed low and passed it on. And to cut a long story short, after 3 generations, that is indeed what has happened. The lines have diverged. They are on average 11% different in their methane emissions, and in 2019 the top and the bottom ram were over 20% different in their enteric methane breeding value. So we successfully know that we can pass it on to the next generation, and that we can breed for this trait. So breeding for something is all very fine, but of course we expect it to have consequences, and the methane animals, or the lines that were measured through respiration chambers, were also measured for a number of other things to see what impact we were having on the animal when we were breeding for this low enteric emissions. So they were measured using various measurement technologies. Their genomes were profiled. They were put through CT scanners to see what was happening to their alimentary tract. Stomach tubing samples were taken to look at the microbes in the gut and what was happening to those. Their feeding patterns were looked at, how often they fed and how much they ate, the carcass and the meat and the quality, and also the reproductive status, the survival, and the disease status of these animals was all looked at in detail. And the resulting program, after many years of work, found that indeed after 3 generations we have 11% less methane per kilogram of feed eaten. So there's no point just breeding for low methane, because what you may breed is a smaller sheep. What we wanted is less methane per feed input. So we have 11% less in the low line. They have a different alimentary tract, so they have small rumens. They have more papillae within that rumen, so they have the same absorptive capacity, but they are indeed smaller. They have different microbes in their guts, so we can detect the difference between the high and the lows by taking a sample of the microbes. They have a slightly different energy profile, so this microbial breakdown of feed is producing a different ratio of the volatile fatty acids, or the energy for the animal, and that's resulting in a different fatty acid profile in the milk and the meat. So we've bred something that's different from its high emitting counterpart. But importantly, if we look at a maternal worth index, so this is in cents, and this is the gross margin or cents per breeding ewe. The orange line is an elite line. It's a maternal flock that's producing really well. The blue line is the average in the nation. The green line is the low line, and the red line is the high line. And what we can see here is that if we look along the years and we look at the sense of gross margin, and this is additional gross margin to the national average, that actually the low methane animals are doing really, really well. They're up with the elite flocks. The red line, the high emitting animals are pretty average, but the low methane animals are actually producing really well. And if we look at the graph, we can see that those additional scents are a combination of a bit faster lamb growth, more lean meat yield. The adults are slightly smaller, they're less fat. There's better parasite resistance in the low line, and most significantly, they grow more wool. So we have a combination of factors which basically show us that low line is healthy and productive. So that's what we came to with the science. Low methane was passed to the next generation. We could breed for it. There were no detrimental side effects, so we felt that we could roll it out further into the national flock. When we looked at the potential to lower emissions, we were seeing around 1% per year, but that's every year. It's cumulative. And we felt that this was an accountable and measurable method to reduce methane emissions, and the intention was that it would be attributable at the farm gate, so breeders would have the opportunity to take this up themselves and be able to have an impact on their own farm. So we had to move from research, which is, we have technical instrumentation, we can measure many factors, onto the farm. And the biggest and the first step for this was to come up with a way of measuring animals on farm that didn't involve a respiration chamber. A respiration chamber is a 48-hour measure. It's very expensive. There's only one set in the country, and we wanted to be able to do something that was lower cost, that was higher throughput, but that was representative of those respiration chamber measures. And we borrowed an idea from our Australian neighbours. They'd been using a smaller portable chamber to measure methane, and our engineering department came up with a sort of Mark II version of this. And these are basically aluminium chambers. They're sealed. There's a fixed volume of air. There's an extraction fan system which cleans the air completely, and we pop a sheep in here for around about 40 minutes. So we have a valve on top of there with a machine that you might take down a mine to measure gas. And when the sheep goes in, we check that all the gases are as they should be, or zero for methane. We measure carbon dioxide, and we measure the oxygen content in the chamber, and then we measure it again at 20 minutes, and we measure it again at 40 minutes. And the difference between the 40-minute and the zero is basically used then, those moles of gas are used to estimate grams a day for the sheep. And we measure them twice. We measure them once for an hour, and then we measure them 2 weeks later, another independent measure for an hour. And that's a really, really good indication of the lifetime methane emission in grams of the sheep. So it's a simple process, but in order to roll it out even further, we needed to put it on wheels, and that's what we did. So we took these units, we took these respiration chambers, which we had a bank in the research farm, and the engineering team again mounted them onto a trailer so as we could roll up anybody's driveway, and we could measure sheep anywhere in the country. And that was the first and the biggest part really to the rollout. But it was fine to have this and to be able to measure breeders' sheep, to go out and measure someone's sheep, but what are they going to do with that information, and how on earth are they going to use it? The next thing was to give breeders a ranking, to give them a number that was meaningful, that they could compare their sheep. And the way we did that was to take all the information that we gained from the research animals, so the variation that we saw, the heritability, the correlations with other traits, any genomic profiles, and to mount it all into the beef and lamb genetics database. So the idea was that it's all in the national repository. All this data is there, it's ready to be used, and that information was used to create models, to create breeding values, or the worth of an animal in terms of what it will pass on to the next generation for methane. And this was basically put into the national system that's used for all other traits. So we developed a methane breeding value that could be delivered to breeders alongside everything else that they do. So not only now do they get a breeding worth for their animals for production and for health and all the other traits that they normally do, they can also have a methane emission breeding value. So the important step there is that it's delivered in exactly the same way as the other traits are. There's nothing new, there's nothing different, and it can be added on. So that gave us the ability to rank any animal and to provide a breeding value, or for beef and lamb New Zealand to provide a breeding value for any animal that was measured. So any farm that chose to have the trailer up in their shed would get a breeding value. But of course, there are 19 million breeding ewes in the country. We're not going to measure in every animal. It's impossible and it's impractical. So we need to share the load. We need to work out how we can roll that out even further. And the way we do that with other traits and with this one is to use genomic prediction. And that's where we measure representative animals. And then we use a DNA profile for the animals that are closely related to them to estimate what their methane emissions would be according to according to how they relate to the animals that have been measured. And to do that, we need linkages. So we need to measure a representation of animals across farms, and we need RAMs to be used across the national flock and across farms. So In order to get this rollout to work, not only do we need to measure animals on farm, we need to make sure that these animals have a tissue sample taken, they have a DNA profile taken, and we estimated, given the heritability of the trait and the structure of the industry, that we would need around 10,000 measures in the database to create an accurate breeding value. So the breeding value needs to be accurate enough that when you use it, you make progress. So we came up with this number of 10,000, and that's actually really quite doable. So our aim for the next few years is to measure 2,500 animals per year that are representative of the national flock, and to make sure that they have good records and good genotypes. And the way we've done this is that we've teamed up with the PGGRC, the Pastoral Greenhouse Gas Research Consortium, and Beef and Lamb New Zealand Genetics to provide a collaborative approach in the form of an industry support package. And what that means is that when a breeder logs on to a website, www.methanebv.co.nz, they get presented with details on how to treat their animals, how they might have them measured. There's a form that they can fill in where they get contacted, and someone from AgResearch will call them and help them through the process, and eventually, if they want, drive the trailer up their driveway and measure their sheep. Once that process is done, we will pass the information over in the right format to Beef and Lamb, who will produce the breeding value using the normal streams of communication to that breeder. Now, where the PGGRC come in is because we really need that database to be representative, and we need it to help the whole of the national flock. Anybody who has good records and has a DNA profile can apply for an offset, so a support package where they pay for half of the measure. So they pay for the first measure, and the PGGRC will support them in paying for the second measure, so that everybody benefits, so that we actually get out there and we get the resource that we need to make sure that it's rolled out in an accurate way. And that's gone really well. We've had really good uptake. This is a map of the farms that have taken up so far. I have to say a huge thank you to the studs and the breeders that have come on board so far and been really helpful. Been really helpful in helping us design the protocols in our communications, in helping us with technical things. You know, making sure that those technical notes are really applicable. Even schools, Lime Hill School, have actually used our equipment to measure their school sheep, to show the school children how it works, and it's been a really great collaborative program to work on. So I'm just going to take a step back now. I'm going to show you a slide that I presented at the World Congress for Genetics in Livestock Production, and I presented this in Canada 6 years ago. So this is me. I hadn't actually been in New Zealand very long, talking to my colleagues, geneticists across the world, about what we were going to do and how we were going to produce lower methane emitting sheep in New Zealand. And this is to show that it was really a structured plan right from the start. These high and low selection lines from sheep that were representative across the nation were going to be measured through respiration chambers, so a very expensive, very technical process. Then we were going to look at feed level and quality. We were going to measure the carcass and elementary tract, and when this discovery set was done, we would test rapid measures. We looked at portable accumulation chambers, and we looked at other ways too. We put the selection lines and progeny tests through first, and then we included commercial animals right back then. And again, many thanks to the breeders that took part. We DNA profiled the research animals to start the prediction source, to seed the models that we would then feed into the national database, into Sheep Improvement Limited, so for beef and lamb to provide breeding values. And we came up with this commercial animal, this low methane emitting sheep. And what that seeded was a whole heap of global interest. And the Global Research Alliance are funding a project at the moment called Grass to Gas, which we're a part of. We exported chambers to Ireland. So they have a trailer exactly the same as the one in New Zealand, and they're now measuring sheep on Irish research farms. We exported the units to Norway, and they put these units into a beautifully heated Mercedes truck, which I'm reliably informed has a very good coffee machine. So the Norwegians are going around looking at the Norwegian white sheep and measuring their animals. There's a lower tech version in Uruguay. There's a higher tech version in Scotland. The Australians are using gas measures. And basically what we have is we have a national rollout in New Zealand where we've hit the ground on farm, but that is seeding more research around the globe, and eventually we'll combine that together and we'll get a global outlook of what the impact of this research and how we might lower methane emissions in sheep across the world will be. So that's really exciting. And finally, the next steps for us in New Zealand, well, we'll continue to work on this genomic prediction. We'll continue to work on this rollout, on accurate breeding values for sheep breeders, on getting it across the national flock, and on giving breeders the opportunity to have a value for every ram that they use. So as they can then present the cohort of rams that they use and say, this is my methane status, this is what I'm breeding, and this is the status of the animals that I'm using. We'll go one step further. I spoke about the microbes. I spoke about the microbiomes and the fact that when we take a sample from the high and low sheep, we can predict from the microbes in the gut whether an animal is a high or low emitting. So those microbial predictors can be used in sheep on farm, and we're taking samples to work out what the best prediction might be. But they can also be used not only in sheep, but in cattle, in beef cattle, in dairy cattle, in goats. So it's about those technologies and actually transferring it into other industries. Sheep produce a third of the methane in New Zealand. So really what we want is the technologies that we develop in sheep to be applicable across the other 2/3. And one of the ways we can do that is to look at microbes. We're looking at the milk fatty acid differences between the high and low lines. And there are to know is if those differences could be turned around, and rather than an effect of breeding for low methane, be predictive of methane, and if they are, whether we could transfer that to the cattle industry. So we're working really closely with the cattle guys to say, any results that we have, can we transfer them, will they be useful? And finally, from a research perspective now, we're moving to additivity of strategies. We know that we have the breeding in place. We want to know if we add the feeding research that's been going on, the vaccine research, the management research, if we put it into a farm systems sort of context, what happens? What can we actually achieve? How fast can we go? And what can we really do on farm for environmental impact? And one thing I will say is that we've actually implemented breeding for low methane in our elite research flock. We've looked at what happens when we select on methane, and when we put quite a big amount of selection pressure on methane, what does that do? And what we're finding is that it lowers production very slightly, but it has a huge impact on the physical change in methane. So we're working through those economic drivers, those offsets, and really seeing what the future holds and how we can keep pushing the boundaries with the research flocks and demonstrate to breeders so that they know what's ahead of them and what they can expect to achieve using these systems. So I'll leave it there, and I will just thank, it's been a huge body of work, as I've said, across a decade, many people involved, many funding industries or funders and breeders. So again, huge thank you to all those involved.
Thanks, Suzanne. Just a couple of questions coming in. I know we're short on time, but I'll try and sum up some of them. Question that's come in really: do you think animal breeding, is it more host or is it more microbiome?
I think it's a combination of the two, obviously, and what we're looking at in the microbiome is the proportion that's controlled by the host and the host environment, so that they're almost inextricable. But we are seeing that if we combine the two, if we take host information and microbe information, that we get a sort of holistic picture of what's going on and a good prediction.
Yeah. Some more questions in around how sensitive is the methane measurement assay, both, experimental and on farm. And I guess that leads to the sort of next question. How much do you think you can breed? How far can you go when it comes to animal breeding in terms of methane emissions?
So the equipment is very sensitive. We sort of calibrate using thousands of parts per million, and we can detect anywhere from 0 to 100% in methane. We also look at CO2 and oxygen. It's less accurate in the chamber than it would be in a respiration chamber, but yeah, it's a very sensitive measure, and it seems to be performing really well. And in terms of what we think we can achieve, we don't honestly know. We are very confident that we could sort of get to somewhere like 10 to 20% below the national average at the moment, over a number of generations. But where that sort of variation takes us and how long we can select for, we honestly don't know yet.
Yeah, and just one quick question. Have you done any work in terms of the quality of the meat that you're getting from these high-low methane flocks?
So currently signals are good. There's more branch chain fatty acids, and the fat deposition is favourable. We have had one small taste test, and actually nobody could tell the difference between the lines. But in general, we haven't seen anything detrimental.
Brilliant. Okay, everyone, I'm really conscious we've gone over a little bit of time, so I think we'll have to end it there. As we said at the beginning, the NZAGRC will be following up with all of the speakers to get answers to the questions that have been asked. I just finally like to finish up by saying thank you to all of our speakers for their contribution today. It's very much appreciated. I'd also like to thank everybody for tuning in this morning. Thanks for your great questions and making it so interactive. And just a quick note, this is the 5th webinar. There's one more to go. So if you're interested, please check out our website and register for that next week's webinar. Same time, same place, same bat channel, so to speak. So again, we'll leave it at that. Thank you, everybody, and goodbye for now.